phosphatase osteogenic marker is highly increased in these cells. Here you can see the adipogenic differentiation of the cells, and here the glucosamine glycans expression when the chondrogenic differentiation is induced. So far, just to conclude, we can say that MECs can be immunoisolated from bone marrow and adipose tissue samples, and we can say that these stem cells are actually multipotent, and the more attractive finding was that we can use these cells without culturing them for in vivo applications. If we look at the rationale behind using mesenchymal stem cells, for example, for bone tissue engineering, so this clinical condition where this long bone was totally crushed due to trauma, this kind of injuries, unfortunately, they do not heal by themselves, and usually they need surgical intervention using metallic wires. This is actually the current available treatment, and as you can see here, this is the metallic prosthesis inserted into this long bone in order to help the healing. And just to see some figures, each year in the United States there's 1 million complicated fractures that is associated with major bone loss, such as this injury, and 1.5 million osteoporosis-related fractures also occur each year in the United States. Unfortunately, these kind of treatments are actually fail in the long term, and it demands many surgical interventions. So in order to overcome this kind of injuries, we first developed a segmental defect in the radius of mice, and this was proven to be a non-healing defect. That means if you do not intervene with these defects, there is no healing. And when we implanted only mesenchymal stem cells that were engineered with a reporter gene, we could not see any healing at this group. These are the micro-CT three-dimensional scans. Here are the cross-sectional scans. But when we implanted at the site of injury mesenchymal stem cells expressing the BMP2 gene, we could see a very nice bridging of the fracture. And as you can see, this newly formed bone quite resembles the original bone. We further went on with this experiment, and we were interested to see whether this bone that was formed by the mesenchymal stem cells expressing the BMP2, whether it is can undergo normal remodeling like native bone, and whether it can actually function from the biomechanical aspect. So we followed by micro-CT scans this bone for 10 weeks and 35 weeks following the implantation, and we could see that this bone actually remained at the site of injury. And when we tested when we tested first we tested the bone volume that is formed at the injury site. This is the group of the mesenchymal, the implanted cells with the BMP2, and we compared this group to the native bone of the mouse. We could see that there was actually some overgrowth of the bone that was formed by the MSCs, but with time, this is after 35 weeks of implantation, we could see that actually the size of the bone is decreasing and becoming more similar to the native bone. If we look at the bone mineral density, we can see that at 35 weeks, actually the bone mineral density of the newly formed bone quite resembles the native bone. If we look at the function of this newly formed bone, we can see that the level of trabeculation is much higher at the beginning, but as remodeling proceeds, this feature becomes more similar to the native bone. And here you can see that also the stiffness of this bone is resembling the native bone. One of the limiting factors that we realized after the implantation of the cells at the injury site was that the cells suffer a lot due to the oxygen depletion and the lack of oxygen that is found at the injury site. And actually, when you implant the cells, most of the cells die due to this oxygen deprivation. So in this experiment, we try to enrich the injury site with oxygen by using synthetic oxygen carriers, that is the PFTBA. And if we measure at ectopic site the volume of bone that is formed following the implantation of the engineered cells, you can see significantly higher bone volume that is achieved when the injury site is enriched with oxygen. This was also shown in an orthotopic, what we call orthotopic site, that is the radius defect model. And you can see here again that the enrichment with oxygen highly increased the bone volume and the bone mineral density. So just to summarize till now, we can say that engineered MCs regenerate bone and repair critical size bone defects in vivo. Repaired long bones remodels and maintains functional biomechanical properties over time. And synthetic oxygen carriers enhance MSC survival and bone tissue formation also in vivo. Another modality that we used stem cells, mesenchymal stem cells, in order to treat was in the spinal disorders. And in most of the spinal disorders that are related to aging actually involve the intervertebral disc degeneration that occurs with age, and this usually causes a very a lot of what is called the low back pain, and that is related to functional limitations of the patients. One of the current approaches, surgical approaches to repair this clinical condition, is to induce the fusion of two adjacent vertebrae in order to limit the movement of this area. And as you can see here, this can be achieved surgically using metallic devices.